Okay, hello everyone, and welcome to BrainMap. It is my great pleasure to introduce our speaker today, Dr. Nicholas Hubbard. Dr. Hubbard is the director of the Neurocognitive Translation Lab at the Center for Brain Biology and Behavior, and an assistant professor of psychology at the University of Nebraska. His lab is the intersection of cognitive neuroscience, clinical psychology, and biomedical engineering, and his research applies novel neuroimaging and statistical techniques to better understand the continuum of mental abilities as they manifest throughout development and in clinical populations. I would just like to remind our audience to please address any question you have using the Q&A box. Dr. Hubbard, thank you very much for coming here today. The virtual stage is all yours.
Oh, thank you so much, Or, and thank you all for coming and for this wonderful invitation. So I am going to turn off my video so you guys don't have to look at me, but I presume you can still see my screen. So this afternoon, I'm going to discuss some of my works that really strive to ascertain more physiologically specific brain signals, and particularly I'll discuss a calibrated magnetic resonance imaging approach that we've used in my lab. So in outline of today's discussion, for many of you this will probably be an overview, but for some it might be a little bit new, so I'll just quickly give a primer on fMRI activation and its relationship to neurophysiological processes. And then I'll discuss the use of calibration fMRI signals for measuring activations in specific neurophysiological compartments. And I'll show some of my work examining the feasibility of this approach and some advantages in one patient population. And finally, I will discuss using calibrated fMRI to examine the organizational properties of oxygen metabolism during the human resting state. Lastly, I'll discuss some challenges and pathways forward for prospective users of calibrated fMRI approaches. So what are we referring to when we say something vague like fMRI activation or brain activation? Well, really what we're talking about is a confluence of several nearly simultaneous physiologic processes. So when we stimulate some population of neurons, our now active neurons, they increase their oxygen use, or they increase their oxygen metabolism. This oxygen is brought to these regions, to these neurons through oxyhemoglobin, and these oxygen molecules are removed from local oxyhemoglobin, and thus converting this into deoxygenated hemoglobin. After this neural activity occurs, our oxygen supplies are replenished by sending oxyhemoglobin through to these local active sites through neurovasculature. But we tend to have somewhat of an overcompensation. Our neurovasculature brings a lot of oxyhemoglobin, which yields a net decrease in local deoxyhemoglobin concentrations. So this is an example using optical imaging of hemoglobin species, so oxyhemoglobin and deoxyhemoglobin within rat barrel cortex after whisker stimulation. So we have our stimulus, and we can see the relative concentrations of oxyhemoglobin and deoxyhemoglobin post-stimulation, where we get this massive influx of oxyhemoglobin and a relatively large efflux of deoxyhemoglobin leaving the local areas. So for our most commonly used MRI activation measure, BOLD, blood oxygen level dependent signaling, it's highly contingent upon this process. So our neurovasculature brings a lot of oxyhemoglobin to the area, resulting in net decreases in local deoxyhemoglobin concentrations and increases in our standard blood oxygen level dependent signal after some stimulation over some time period. So with this primer in mind, what does this standard bold contrast tell us? Well, it can yield a very excellent marker of where and when broad physiological changes are occurring associated with, say, some stimulus. So we can look at V1 bold signal given some flicker frequency task, and we know that our increases and decreases in bold signal are corresponding relatively well with our task. But we also can see where this is happening. So say looking in V1 itself. But because bold is a confluence of blood flow, neural activation, and neurovascular signaling processes, we don't really know which of these physiological compartments give rise to these bold signal changes that we're seeing. So in the typical healthy brain, relatively tight coupling between physiological responses across these compartments can allow for reasonable inferences to be made that our bold signal changes that we're observing are reflecting signal changes in our neuronal compartment, so reflecting neural activation. However, departures from this normal physiology, such as in altered physiological states like pathology or pharmaceutical manipulation, this inference is relatively tenuous. So for example, caffeine is a relatively potent vasoconstrictor, and about 2 cups of coffee or 200 milligrams of caffeine is related to an uncoupling of our neural compartment or neural activation from bold signal. In even states, relatively healthy states, but still slightly different physiological states like healthy aging, have been shown to be related to an uncoupling of our neural activation from bold signal. So emerging fMRI approaches like calibrated fMRI, which is the technique that I'll discuss today, they can target the where, when, but also what are the specific neurophysiological changes that are occurring associated with, say, some stimulus. So calibrated fMRI uses our standard bold signal and blood flow or volume signals along with a biophysical modeling approach to estimate changes in these different physiological compartments over time. So it can allow us to estimate oxygen metabolism changes reflecting our neuronal compartment, neurovascular coupling changes reflecting our neurovascular signaling compartment, as well as blood flow, which are going to reflect our vascular compartment. And some potential advantages of this technique, at least that I see, are it can allow us to identify the physiological responses, say, related to healthy and pathological neural activation, as well as understanding the neurophysiological bases of pathological or some type of interventional change, like through a drug study. So just a quick digression to support the idea that oxygen metabolism and calibrated oxygen metabolism measures specifically are a good proxy for measuring activation in our neuronal compartment. Well, oxygen metabolism is required to support neuronal activation and is intimately linked to signatures of this activation. So during stimulation, about 98% of neuronal ATP, adenosine triphosphate regeneration, which is our main metabolic substrate, occurs due to oxidative metabolism. There's a near perfect relationship between calibrated imaging measures of oxygen metabolism and postsynaptic spiking potentials and glutamate cycling, so relationships between these electrochemical signatures of neural activation. So this is just a study showing changes in the calibration-derived oxygen metabolism signal in rat somatosensory cortex during 4-pulse stimulation. And you can see that at least in time, these correspond very, very well to measures of changes in postsynaptic spiking potentials. And if we want to talk about our calibrated oxygen metabolism measure relative to our current field standard of BOLD, well, calibration-derived oxygen metabolism shows about 2 times greater predictive ability than BOLD for local field potentials, and about 8 times greater predictive ability than BOLD for multi-unit activity. So both our measures of pre- and postsynaptic neural signaling show a stronger relationship with calibration-derived oxygen metabolism measures than they do with BOLD. All right, so how do we get our measurements of oxygen metabolism and these other compartments via calibrated imaging? So I'll discuss a relatively typical calibration approach, which really has 2 steps. So it's very common for folks to acquire near simultaneous BOLD and arterial spin labeling signals, say using a dual echo approach during a task state, and also applying this same dual echo approach during a resting physiological challenge. When we talk about dual echoes, we'd say maybe our first echo is going to reflect the label or control from P-Castle, whereas our second echo would reflect BOLD weighted signal. And the second step is to extract signal changes acquired during the physiological challenge to calibrate the task state BOLD activations. And this is how we acquire oxygen metabolism measures, as well as our other estimates of these physiological compartments. So probably the most common physiological challenge involves breathing room air and for several minutes in, say, a hypercapnic or CO2 heavy solution for another several minutes afterwards. So this calibration factor that is required to apply to our task states is acquired through this hypercapnic challenge. So CO2 is a very potent vasodilator. So it increases global blood flow, and this increase in blood flow, but relatively negligible increase, if any, in baseline deoxyhemoglobin, maximally dilutes the baseline deoxyhemoglobin concentrations, the local deoxyhemoglobin concentrations, and this allows for the estimation of the influence of maximum changes in deoxyhemoglobin concentrations on our BOLD signal from baseline. So what we end up with is a maximum BOLD estimate, which allows us to scale our target BOLD signal via this biophysical modeling approach. And so here is just an example from one of my studies where we're showing the average maximum BOLD signal change, or M, acquired through a hypercapnic challenge across 15 healthy young participants. So the biophysical model approach that we often use is the deoxyhemoglobin dilution model of BOLD signal. So this is used to derive evoked changes in the cerebral metabolic rate of oxygen. So I'm really only going to describe the CMR02 portion of this model, because activation in the other compartments are either acquired or calculated using some derivative of CMR02. So essentially we have our BOLD signal changes that are occurring during our task. which are scaled by this calibration factor, which is acquired during a physiological challenge, as well as we look at our blood flow changes that also are occurring during the task to acquire task-related changes in the cerebral metabolic rate of oxygen. And there are also some empirical constants that are assumed as well within this deoxyhemoglobin dilution level. So my works using calibrated fMRI have sought to test the feasibility of this method for use in patient groups, as well as to test whether examining these additional compartments using what is obviously a much more complicated approach, can this provide any useful information about these groups' pathophysiological processes, say above and beyond bold signal. So the studies I'm going to talk about today look at the relationships between white matter damage in multiple sclerosis and evoked neurophysiological responses. So for those of you who might not be familiar with MS, MS is an autoimmune disorder characterized by gross white matter lesions and microstructural damage, as well as heterogeneous changes to neurometabolism and general decreases in blood flow. So the reason I sought to examine multiple sclerosis is previous work that I had also done showed differences in evoked responses between MS patients compared to controls. And these differences were essentially mediated by the extent to which each patient had some form of white matter microstructural damage. So in the first study that I'm going to discuss, we sought to ask, well, which physiological compartments does this MS-related white matter damage? What does it affect? So here we use dual echo, the dual echo calibration approach described above during visual stimulation, along with diffusion tensor analyses of occipital white matter tracks in there. The integrity of the white matter microstructure in these tracks, and look at relationships between, say, bold cerebral blood flow and the cerebral metabolic rate of oxygen in visual cortex, how they're associated with these various white matter tract integrities. So what we had found was that greater white matter microstructural damage, so increased here on the, on the x-axis, looking at the occipital tract radial diffusivity from our diffusion tensor imaging, was related to greater evoked visual cortex oxygen metabolism. And what was important about this is this correlation between microstructural damage and evoked oxygen metabolism was much stronger than any other measured variable that we had looked at. So whether using bold cerebral blood flow image evoked measures, or looking at things like gross lesion burden or gross gray matter atrophy. So we also sought to test whether CMR02 could potentially provide any unique information about our patient's disease burden. White matter microstructural damage is related to their disease burden after all. So we were curious to see if looking at this calibration-derived oxygen metabolism had any relationship as well to disease burden in multiple sclerosis. So what we had found was, again, greater visually evoked oxygen metabolism, now on the x-axis, significantly and uniquely accounted for MS patients' extent of clinician-rated neurological disability. And this relationship, importantly, remains significant even when controlling for other MS-related factors, such as white matter microstructural damage, gross white matter damage, looking at lesion burden, as well as looking at things like gray matter and white matter atrophies. We also found that this greater evoked oxygen metabolism measure, as well, along with blood flow, our cerebral blood flow measure. Both of these were related to greater fatigue symptoms, self-reported fatigue symptoms in multiple sclerosis. And for those of you who are familiar with MS, you might be aware that fatigue happens to be a primary complaint in multiple sclerosis. So with this, we found some evidence of CMR02 relating to disease burden as well as white matter microstructural damage. But I also wanted to see whether or not this visually evoked oxygen metabolism measure, whether this could have some type of diagnostic utility. So in a second study, we asked the question is, can visually evoked oxygen metabolism, could this provide some type of putative diagnostic marker of MS? So here we repeated the same procedures as mentioned in the previous study with additional age and sex-matched controls. And we examined visually evoked oxygen metabolism, blood flow, BOLD, as well as about 39 or 38 or so other MS-related neuropsychological, imaging, and self-report variables. So compared to our other imaging measures, visually evoked oxygen metabolism was the strongest physiological predictor, strongest activational, we could say, predictor of MS status. So we had about 82% in sample accuracy and about 77% cross-validated accuracy. And here you can see the classification accuracy of MS status as well as the effect sizes associated with visually evoked oxygen metabolism, visually evoked cerebral blood flow, BOLD, as well as a measure of N, which is our measure of neurovascular signaling. So through this, and as I mentioned, in that previous study, we had looked at multiple other measures that were previously related to multiple sclerosis pathology in one way or another. And what we had found was visually evoked oxygen metabolism was within, I think, the top 15% of all predictors for an MS diagnosis. So through these studies of multiple sclerosis, we were able to newly employ calibrated fMRI in a neurological population, which had never been done before, as well as newly combine diffusion-weighted imaging along with calibrated imaging. Again, had never been done, and to my knowledge, I don't think it's been done since. So what we'd found was relatively clear advantages for the use of calibration-derived oxygen metabolism measure relative to BOLD, to some extent blood flow, and various other imaging and neuropsychological measures for examining MS-related white matter damage as well as their primary symptoms. So with this, we were able to really provide a proof of concept for the application of calibrated imaging in a pretty severe patient group. And together, these findings suggest optimism for examining this visually evoked oxygen metabolism measure as a potential prognostic or diagnostic marker of multiple sclerosis. I mean, this said, these are very preliminary works with few subjects and a cross-sectional design, which certainly has implications for prognoses. So I view these studies as quite preliminary in the hopes that more work could be generated from these findings as well. So far, we've discussed the use of calibrated fMRI to examine the stimulated or active states of our brain. However, these really represent an exceedingly small fraction of our brain's functions. So for instance, about 85 to 95% of our brain's total energy use occurs when the brain is not currently engaged by exogenous stimuli, so things like our resting state. And most of these resting neuronal processes are maintained by oxidative metabolism, so oxidation of glucose, about 98% of these. And our intrinsic communication networks of the brain have been shown to be almost entirely maintained by glucose oxidation. So looking at, say, resting communication networks, these are very strongly sort of influenced by the extent of glucose oxidation. So here, from one work, we see ATP turnover on the y-axis and various resting state networks on the x-axis. And in the very small open bars, we see the amount of sort of energetic turnover by glycolysis relative to the total, which the difference would be that maintained by glucose oxidation. So this difference is relatively large, implying that these communication networks or subnetworks of the human brain are indeed almost entirely maintained by glucose oxidation. So naturally, because the resting brain and sort of its intrinsic networks and intrinsic organizational properties are so dependent upon oxygen metabolism, I was really curious to see if our calibration-derived measure of CMRO2, could this be used to examine the general organizational properties of the resting brain? So the question that I'm going to discuss in a paper that we have currently under review is really, can calibration-derived oxygen metabolism signals, can these detect typical patterns of brain organization? So in this study, we had 15 participants that underwent dual echo imaging during a 20-minute sort of typical resting fixation period, as well as a 10-minute hypercapnic challenge. We looked at low frequency fluctuations of oxygen metabolism by applying a dynamic adaptation of the deoxyhemoglobin dilution model to these near simultaneously acquired resting state fluctuations of BOLD and blood flow signals. What we tested was whether the interregional correlations between low frequency fluctuations of oxygen metabolism could produce organizational signatures consistent with previous findings of brain structural and functional connectivities. So the first question that I sought to examine was whether the topological properties of, say, an oxygen metabolism network were consistent with previous findings from structural and functional imaging. So generally, our brain connectivity shows some form of complex or small world topology, meaning that it demonstrates relatively high tendencies for proximal nodes in the network to be connected with one another, this idea thought of as segregation, as well as relatively short path lengths between each node within the network. So this idea operationalized as integration. So our brain topology, how we operationalize these tests of sort of small worldness or complexity, is by examining whether or not, say, a brain measure's topology demonstrates greater segregation properties than similarly sized and dense random networks, and greater integration properties than completely ordered networks that are of similar sizes and densities. So these are where only the nearest neighbor nodes are connected to one another. And these are also referred to as lattice networks. So it might look, a diagram of this might look something like this, where a small world network, which in this case we're assuming is our brain topology, would show greater segregation properties than a random network and greater integration properties than a lattice network, and would show higher complexity relative to both. So after recovering our CMRO2 resting fluctuations using this dynamic adaptation of the deoxyhemoglobin dilution model, our next task was to create an oxygen metabolism network. And to do this, we segmented the brain into discrete nodes based on the correlations, sort of spatially constrained correlations between low frequency fluctuations of oxygen metabolism. So the approach we use, some folks might be familiar with, was often used in fold-based studies where our nodes were created from spatially constrained spectral clustering of CMRO2 low frequency fluctuations. And what we looked at was to compare the oxygen metabolism network, its segregation and integration properties, to around 7,500 simulations of random and lattice-like networks, which use approximately the same number of nodes. So actually the exact number of nodes with 194 and very similar number of connections. So consistent with previous findings of anatomical and functional connectivity, the OMNs indeed demonstrated topological properties generally indicative of complex or small world networks. That is, when we look at the distribution of segregation coefficients, or C, or clustering coefficients, C, for oxygen metabolism, we see that they're much greater than similarly sized and dense random network simulations. And when we look at this, the integration properties, looking at the inverse path length measure, we see that our oxygen metabolism networks were indeed much greater than similarly sized and dense lattice-like networks. And I should mention we use the term lattice-like instead of lattice networks because a true lattice network can't really be simulated with the exact equal size and density of all of the OMNs that we had looked at. And so this is just a biplot demonstrating the individual networks at multiple different correlation thresholds for our different network types. So for our oxygen metabolism network, our lattice-like networks, as well as our random networks. And we have integration and segregation plotted here. And you can see that, again, we show greater integration properties than a lattice-like network and greater segregation properties than random networks. Just an example of what this might look like for a randomly selected participant. We have one of their oxygen, their random simulations of their oxygen metabolism network, their actual oxygen metabolism network, as well as their lattice-like networks. And these generally fall in between these random and lattice-like networks, indicating relatively high complexity and relatively small world-esque topology. So just to show parity of this for our other measures, our other signal measures, so across both cerebral blood flow and oxygen metabolism network topologies, these generally demonstrate similar small world properties when compared to their respective simulated networks. So essentially the distance between these circles and the square or of the triangles of the same color are relatively equidistant. So aside from examining sort of the global network topology of oxygen metabolism, we also sought to examine whether calibration-derived oxygen metabolism could demonstrate subnetwork connectivity patterns demonstrated previously by BOLD and cerebral blood flow measures. So here we looked at 4 different resting state. I call them subnetworks just so they're not confused with the overall oxygen metabolism network that most folks would call these networks. We looked at 4 different of these subnetworks that have been shown to be relatively reliable and detected in both bold and cerebral blood flow studies. So we looked at the default mode network, occipital visual network, auditory network, as well as the right and left frontal parietal networks combined into just a frontal parietal network. So here we essentially used a seed-based approach to recover default mode, occipital visual, auditory, and frontal parietal subnetworks. And we tested the relationship between the oxygen metabolism correlation patterns with these individual subnetwork seeds and the correlation patterns derived from an open database of resting state bold images of 1,000 participants. So we use NeuroSynth for those folks who are familiar with NeuroSynth. So just a quick view of what the recovered networks looked like from our measures and from NeuroSynth. So our measure is on top, and this is for the default mode network seed, frontal parietal network seeds, occipital visual network seed, as well as the auditory network seed. So on top, we have our calibration-derived oxygen metabolism correlations with the network seed as well. And on the bottom, we have the ones derived from the resting sample of 1,000 participants using bold. So qualitatively, these look relatively similar, although also relatively diminished for the calibration-derived oxygen metabolism relative to bold. But quantitatively, we wanted to test the relationships between all of our connectivity measurements. So all of our correlations based on using correlations with the CMR2 based network patterns, as well as the bold 1000 or NeuroSynth subnetwork connectivity patterns. And here we showed relatively large effect size relationships between oxygen metabolism derived subnetwork connectivity patterns and our bold 1000 voxel-wise subnetwork connectivity patterns. Here you can see it for using the default mode network seed, the frontal parietal seeds, the occipital visual seed, as well as the auditory network seeds. We also wanted to test the spatial overlap between thresholded correlation maps. So we tested, we thresholded the oxygen metabolism networks, subnetworks, and bold 1000 subnetworks. We looked at the top 10% of connections and as well as their spatial overlap. And what we found was medium to large effect size relationships, looking at the binary association coefficient phi. So we average around about half of the variability, half of the variance accounted for, or excuse me, 1/4 of the variance accounted for in the bold signal using the CMR2 only approach, which is actually pretty good considering that this is out of sample prediction using a different physiological marker than bold acquired from different makes and models of scanners and processed using completely different techniques as well as different resolutions. So just to give you an idea of what this looks at with a more comparable comparison, although I think these are much more rigorous comparisons, but we also did look at relationships between oxygen metabolism and the acquired dual echo bold, their voxel-wise subnetwork connectivity patterns, which also showed large effect size relationships between these two approaches. These were obviously markedly larger, considering that there were very few differences between the samples. This is something that we would expect. And we also observed enhanced spatial overlap when examining the oxygen metabolism measures of subnetwork connectivity and the dual echo-based bold measures of subnetwork connectivity. So all of these essentially constituting large effect size relationships. So returning to the key question, can calibration-derived oxygen metabolism, can it detect typical patterns of brain organization? Well, what we had found was interregional correlations between the low-frequency fluctuations in calibration-derived oxygen metabolism did indeed demonstrate complex small world properties typical of previous structural and functional connectivity findings. And we also showed this with our dual echo bold in cerebral blood flow signals, that there was parity there. We further demonstrated similar voxel-wise subnetwork connectivity patterns to bold, where we had large effect sizes correlations with the voxel to voxel relationships and medium to large effect size relationships for when comparing the calibration-derived CMRO2 to the neurosynth bold data. And these also were significant when comparing to the dual echo bold images, albeit much greater so. So this is, again, a sort of proof of concept study, but our takeaway from this was that calibration-derived oxygen metabolism can detect typical organizational patterns of brain connectivity. And this does suggest optimism for broader use of this method to target physiological processes intimately linked to intrinsic neuronal communication and intrinsic neuronal activation. So it's really important to note, and I just want to close with this, that calibrated imaging is really in its infancy. And if we'd like to see this method adopted more widely, I think we need to really overcome some few key challenges first. And the first is that calibration-derived oxygen metabolism has a very, very low signal to noise relative to bold and relative to blood flow signals. So currently this means that more oxygen metabolism data are actually needed to achieve the same statistical power relative to cerebral blood flow or bold signals alone. Statistically, noise can be reduced for oxygen metabolism signals via constraining the variability of our blood flow fluctuations to the variability of bold fluctuations. And this has been shown in tasks via the bold constrained perfusion approach, but it's not presently known whether this variability removed is going to be appropriate within the human resting state. A second challenge is that we really need to circumvent the physiological challenges that are part of most calibrated imaging studies. These are simply not viable for some populations and are very difficult to employ relative to standard bold imaging. There is some evidence out there that I think is tentatively provides some tentative optimism for recovering our bold scaling factor, or M, without the use of physiological challenges. But this is still very much experimental, I would say. And finally, calibrated imaging signals have decreased spatial and temporal resolutions relative to our more standard approaches. So dual echo signals are acquired 2 to 8 times slower than the typical sampling rate of our standard bold. And so this does require more scanning time for a comparable number of dynamics. Calibrated imaging measures also show about a 3 millimeter increase in slice thickness for each voxel plane relative to standard bold. And so for a lot of participants, our acquisition simply cannot cover the entire brain of all participants. There is some evidence out there that applying multiband dual echo sequences may indeed be able to remediate this issue. So I just wanted to thank all of my wonderful collaborators from UT Dallas, UT Southwestern Medical Center, Hopkins, UPenn, as well as the Baylor College of Medicine. And thank you all so much for your time and for your attention. And please feel free to let me know if you have questions, comments.